Hello, and welcome to Arcadia University's BI327 Histology Epithelial Tissues lecture. In part one of this lecture, we're going to look at an introduction to epithelial tissues. As always, take a look at the objectives for this series of lectures so you get an idea of what are the important concepts to be getting out of the lectures and the activities as you go through. Now, as we start taking a look at the individual essentially tissues within the body, as we start kind of an overview of this concept of microanatomy, the study of the microscopic anatomy and physiology, it's important to recognize that there are four basic tissue types. Two of the tissue types are grouped together based on morphological or essentially anatomical similarities. The other two were grouped together based on functional similarities, based on physiological similarities. Now, in terms of the morphological or anatomical similarities, the first are the epithelial tissues, which is going to be the purposes of this lecture, where you basically have a closely packed sheet of cells, which in essence are going to be involved with forming a sheet. In contrast, connective tissues, the second type of cells that we're going to take a look at, are cells that are widely scattered throughout an extracellular matrix. So they're not in contact with one another, and they're producing a lot of material that is going to be found between the individual cells. So those are the anatomical characteristics associated with either the epithelial or the connective tissues. The other ways that tissues are grouped together are based on functional or physiological similarities. The first are the nervous tissues, and the nervous tissues are excitable cells, which are capable of generating, receiving, and sending electrical signals, either chemical or electrical signals. And then the other type of tissues are going to be the muscular tissues. The muscular tissues are going to be excitable cells that are capable of, when they receive a signal, of undergoing contraction. And so the contraction is going to be the physiological or functional characteristic associated with all muscle cells. But the purposes of this lecture and the various parts of this lecture are going to be the epithelial cells. So again, if we take a look at this from the very basic characteristics, what we've got with an epithelium is that we're going to have a set of closely packed cells which are going to be forming sheets within the body. So in order to find an epithelium, what you're going to do is basically look for a surface, because the epithelium is going to be lining every surface of the body, inside and outside of the body. So inside of the digestive system, outside of the surface of the body, are all going to be lined by an epithelium, except for articular cartilages, where you've got two bones coming together, and the actually cartilage at the tip of the bones are going to be coming together in joint cavities, is not going to be covered by an epithelium. And it's the only region in the body where you're going to have a free biological surface without an epithelium lining. If you take a look at the overall characteristics again of an epithelium, they're going to be avascular, which means that they don't have a direct blood supply. They're going to be dependent upon diffusion of materials from the surrounding areas. And in general, epithelia are often going to have a very robust nerve supply associated with them. They're going to be very well innervated. If we take a look at this and try to come up with some basic characteristics associated with an epithelium, we're going to see that the general function of an epithelium at the tissue and organ level is going to be very similar to what we saw with the plasma membrane or cell membranes at the cellular level. So they're going to be separating self from non-self. So the epithelium is going to be separating the inside of a structure from the outside of the structure, the same way that the cell membrane separated the inside of the cell from the outside of the cell. Epithelium is going to be involved with dividing the body into functional compartments. They're going to help identify organs and substructures of organs within the body, in the same way that a cell membrane created subcellular structures, subcellular organelles. The epithelium is going to be involved with actively and passively regulating the movement of materials between compartments. And so it's going to form essentially a semi-permeable barrier, which is going to regulate and control and restrict the movement of materials across the epithelium. And when it does that, it's possible that that epithelium may be modifying the substances as it's moving them across this boundary. So if we take a look at this again from the very basic level, what you're going to see if you take a look at take a look at a biological specimen. So you're going to look for a natural space. So you're going to look for an opening in a tissue specimen. And then you're going to see the epithelial lining, these kind of greenish cells on the diagram we've got here. Underlying that epithelial lining, again, the closely packed cells forming a sheet of cells, we're going to have the basement membrane, which is that reddish kind of layer that we're looking at it here. And underlying that, we're going to have the lamina propria. Lamina propria will focus in on more detail when we talk about connective tissue, but this is the supportive connective tissue layer, which is going to be underlying the epithelium. And this is going to be consistent whether you look at different epithelia through the body. If we take a look at that basement membrane in kind of more detail, what we're going to see is that the basement membrane is going to be that boundary between the epithelia and connective tissue, and it's going to be important for integrating the epithelium and connecting it into the surrounding connective tissue. Now, the basement membrane is, in essence, a light microscopy term. If we were to look at it on electron microscopy, what we've got with this diagram on the right-hand side, we're going to see a basal lamina, so essentially a subset of that. But the overall characteristic of the basement membrane is that we're going to have a lot of glycoproteins, essentially protein filaments, which are going to be there for strength and integrity, almost like a little scaffold system, which is going to help us to anchor our epithelium into the connective tissue and provide some structural stability. The glycoproteins in this region are going to be things like collagen, as well as specialized glycoproteins like heparan sulfate proteoglycan, laminin, fibronectin, a whole range of things there. But basically filamentous proteins, fibrous proteins, which are involved with anchoring our epithelial lining to the surrounding tissues. Now, the overall functions associated with the basement membrane are primarily provide attachment sites, protection, and support, but in some regions, it's also going to have an ultrafiltration capacity. We look at what's occurring within the kidney, the basement membrane is going to be contributing to the ability to regulate the flow of materials through the barriers. Under most circumstances, blood vessels are not going to penetrate through the basement membrane. They're not going to penetrate through the basal lamina. And so the epithelium is dependent upon diffusion of materials up through that lamina propria into the epithelium itself. Now, if we take a look at the epithelial lining, what we're going to see is that in most cases, an epithelia is going to be going through a fairly constant turnover, and that we're going to have some undifferentiated stem cells, which are going to be capable of dividing, and when they divide, they're going to provide a new population of cells for the repair or renewal replacement of the existing cells within the epithelium. What this means, though, is that you've got cells that are going to be continually to divide well into adulthood, and so it means that there's a risk for these cells to have disruption of that normal mitosis, that normal cell reproduction. And so because of that, what we see is that most of the malignancies that occur in adulthood are going to be in epithelial nature. And so what we're looking at is either a metaplasia, where we have a change in the epithelial type of cells due to infection or irritation, or a dysplasia, which is an abnormal pattern of growth associated with the production of these cells. Again, taking a look at these epithelial cells and the susceptibility associated with them. If you have any questions on the first part of this lecture, feel free to email me at hoffmanj@arcadia.edu. Thanks.